Hello students. Today we are going to discuss about Unit 5 Transgenic Fish, Zoology BSc third year, semester 6. The objectives of this chapter are to understand about transgenic fishes, to know about transgenic fish production, to understand the importance of transgenic fish in aquaculture. To introduce the topic, organisms into which heterologous has been artificially introduced and integrated in their genomes are called transgenics. Since the early 1980s, transgenic plants, nematodes, fruit flies, sea urchins, frogs, laboratory mice, and farm animals such as cows, pigs, and sheep have been successfully produced. In plants, transgenes are introduced into cells by infection with Acrobacterium tumefidens or by physical means such as ballistic bombardment. In animals, transgenes are introduced into the pronuclei of fertilized eggs by injection, and the injected embryos are incubated in vitro or implanted into the uterus of a pseudo pregnant female for subsequent development. So these transgenes in many transgenic individuals they're transmitted through the germ line to subsequent generations. These transgenic animals play a very important role in basic research as well as applied biotechnology. In basic research, transgenic animals provide excellent models for studying molecular genetics of early vertebrate development, actions of oncogenes, and the biological functions of hormones at different stages of development. In applied biotechnology, transgenic animals offer unique opportunities for producing animal models for biomedical research, improving the genetic background of brood stock for animal husbandry or aquaculture, and designing bioreactors for producing valuable proteins for pharmaceutical or industrial purposes. The transgenic fish is one that contains genes from another species. A transgenic fish is an improved variety of fish provided with one or more desirable foreign genes for the purpose of enhancing fish quality, growth, resistance, and productivity. Since 1985, a wide range of transgenic fish species have been produced by microinjecting or electroporating homologous or heterologous transgenes into newly fertilized or unfertilized eggs. Several Several important steps are routinely taken to produce a desired transgenic fish. Typically, genes of one or more donor species are isolated and spliced into artificially constructed infectious agents, which act as vectors to carry the genes into the cells of recipient species. Once inside a cell, the vector carrying the genes will insert into the cell's genome. The transgenic organism is regenerated from each transformed cell or egg in the case of animals, which has been taken up the foreign genes. And from that organism, a transgenic variety can be bred. In this way, genes can be transferred between distant species which would never interbreed in nature. Aquatic animals are being engineered to increase aquaculture production. The use of genetic engineering and rDNA technology has done miracles in medical and industrial research. The transgenic fish are being promoted as the first marketable transgenic animals for human consumption. One of the most important aspects between fish and other terrestrial animals for cultivation and genetic improvement is that usually fishes have higher levels of genetic variations and hence more scopes for selection than most mammals or birds. Using the gene transfer technology, scientists now have created a genetically engineered variety of Atlantic salmon that grow to market size in about 18 months. Otherwise, the fish takes about 24 to 34 months for becoming market size fish. It is also hoped that we can now modify a large number of fishes with fast growing characteristics and bring blue revolution. The following are the important points needed for genetic engineering, that is, for gene transfer to produce transgenic fish. One, a gene sequence is to isolate for the particular characteristics, for example, growth hormone gene. These genes are then inserted into a circular DNA known as plasmid vector. Enzymes and endonucleases and ligases are used. Then plasmids are harvested in the bacteria to produce billions of copies. Then plasmids are introduced into linear DNA. The linear DNA is sometimes called a gene cassette because it contains several sets of genetic material in addition to new inserted gene. For example, growth hormone gene. The technology is available to integrate genes in germline of developing individual and finally transmitted into further generations, making the cassette permanent part of fish's genetic makeup. Now, how to develop the development of transgenic fishes? Many of the fish under development are being modified to grow faster than their wild or traditionally bred aquaculture siblings. Faster growth is usually accomplished by transferring a fish growth hormone gene from one species of fish into another. The faster growing fish not only reach market size in a shorter time, they also feed more efficiently. Trot growth hormone was used to produce transgenic carp with improved dressing properties. Such transgenic carp are recommended for production in earthen ponds. Let us see transgenic salmon. The Atlantic salmon is engineered with a Pacific salmon, growth hormone driven by the Arctic antifreeze promoter gene. The rapid growth of that transgenic salmon is achieved not so much by the transgenic growth hormone as by the antifreeze gene promoter that functions in the cool water desirable for salmon flavor. The transgenic coho grew on average 11 times faster than unmodified fish, and the largest fish grew 37 times faster. The growth hormone levels in the transgenic fish are high year round rather than falling off in the winter as occurs in ordinary salmon. The modified salmon are large enough to be marketed after 1 year, in contrast to standard farmed salmon that do not reach market size for at least 3 years. Transgenic tilapia. Tilapia fish, which is native to Africa, are cultured worldwide as poor man's food, second only to carp as warm water food fish, and exceeding the production of Atlantic salmon, whose market value is twice that of tilapia. Tilapia has been extensively genetically modified and promoted as a transgenic fish, exclusive for isolated or contained production. Transgenic tilapia, which is modified with pig growth hormone, has 3 times larger than their non transgenic siblings. Tilapia genetically modified with human insulin grew faster. Human insulin grew faster than non transgenic siblings and could also serve as a source of islet cells for transplantation to human subjects. Next, transgenic Medaka fish. Purdue animal scientists Muir and Howard in 1999 used tiny Japanese fish, Oryzias latips, called Medaka, to examine what would happen if male Medakas genetically modified with growth hormone from Atlantic salmon. Inserted a gene construct consisting of the human growth hormone driven by the salmon growth hormone promoter into Medaka, produced the transgenic Medaka we can see in the picture. The availability of groups of modified and conventional fish was measured at 3 days of age, and 30% fewer transgenic fish survived to that age. The researchers calculated that large males had a four-fold mating advantage based on observations of wild type Medaka. In another experiment, silk moth genes were introduced into Medaka fish to create resistance to bacterial pathogens. Then coming to transgenic zebrafish. The tiny zebrafish that lives in aquariums was genetically modified to produce a fluorescent red pigment and is being promoted for sale as a household aquarium pet, the goldfish. The goldfish caused a stir in the United States because regulation of such transgenic pets is murky and none of the major regulatory agencies, Food Drug Administration, United States Department of Agriculture, or Environment Protection Agency, has been willing to take the lead in regulating the goldfish. Gong Gonji developed novel varieties of the zebrafish in the year 2003. Three living color fluorescent proteins, green fluorescent protein, yellow fluorescent protein, red fluorescent protein, were expressed under a strong muscle specific MILS2 promoter in stable lines of transgenic zebrafish. The transgenic zebrafish with vivid fluorescent colors, that is green, yellow, and red, orange, fluorescent proteins can be seen with naked eyes under both daylight and ultraviolet light in dark. The green fluorescent protein is originally isolated from, from the jellyfish. Transgenic common carp. Thomas T. Chen, director of the Biotechnology Center at the University of Connecticut's Stores, transferred into common carp the growth hormone DNA from rainbow trout fused to a sequence from an avian sarcoma virus. The genetic material was injected into fertile carp eggs with microinjection. The offspring of the first generation of transgenic fish grew 20 to 40% faster than their unmodified siblings. Chen is also developing transgenic catfish, tilapia, striped bass, trout, and flounder. Autotransgenesis. Indian scientists are concentrating on developing transgenic fish through autotransgenesis, which involves just increasing the copies of growth hormone genes present in a fish as opposed to allotransgenesis, which amounts to transfer of genes from different species. The increase in growth hormone genes leads to an increase in flesh content. Indian scientists feel that Autotransgenesis is safer and less controversial. According to T.J. Pandian of the School of Biological Sciences in the Madurai Kamaraj University, the generation time of most fish species is shorter and breeding of frequency is relatively higher. A single female can produce several hundred or thousand eggs and thus provide a large number of genetically identical eggs. Besides, the most important advantage is that the fertilization is external and can be rapidly controlled by experimental manipulation. Controlled culture of transgenic fish and feed. Pond commercial culture is effective for carp and tilapia, but more difficult with salmon and trout. Currently, pond culture is suitable for carp and tilapia because the fish are vegetarians. Carnivorous salmon and trout depend on a diet of fish and fish meal, but the worldwide stock of feed fish has diminished and suitable vegetable meat substitute must be found. Gene transfer technology for development of transgenic fishes. The most commonly used methods in fish biotechnology are chromosome manipulation, hormone treatments, which can be produced triploid, tetraploid, haploid, gynogenetic, and androgenetic fish. Other popular methods of gene transfer in fish are microinjection, electroporation of sperms, electroporation of eggs, and incubation of sperms. Let us look at the first one, that is preparation of DNA construct. Desired transgene should be recombinant gene. It should be a recombinant gene or DNA construct, which is constructed in plasmid that contains an appropriate promoter enhancer element and a structural DNA sequence. The foreign genes are typically introduced with strong genetic signals, promoters, and enhancers, which enable the foreign genes to be expressed at very high levels continuously or constitutively, effectively placing those genes outside the normal metabolic regulation of the cell and of the transgenic organism resulting from the transformed cell. There are three main types of transgenes. Gain of function. These transgenes are able to increase particular function in transgenic individual after their expression. For example, growth hormone genes from mammal and fish linked to appropriate promoter enhancer element and a structural DNA sequence to produce GH transgene. This GH transgene, which when expressed in transgenic individuals, increases production of growth hormone, leading to enhanced growth of transgenic animal. Reporter function. These transgenes are able to identify and measure the strength of promoter enhancer element. Then loss of function. This transgene is not yet used for modification of transgenic fish. Such transgenes are used for interfering with the expression of host genes. The promoter enhancer elements of transgenes are linked to a growth hormone gene of fish. Gene transfer by microinjection. Microinjection is most successfully and widely used technique for gene transfer in fish. One method of microinjection technique involves the use of fine injection needle for introducing DNA into cut site in the cell. While doing so, it destroys those cells that are in direct contact with the injected DNA. To ensure the integration of the DNA, it should be injected to intact cells close to the cut site. The injection apparatus consists of a dissecting stereo microscope and two micro manipulators, one with a glass microneedle for delivering transgene and other with a micropipette for holding fish embryo in place. The success of microinjection technique depends on the nature of egg chorion. The soft chorion facilitates the microinjection while the thick chorion limits the ability to visualize the target for injection of DNA. In many fishes, the egg chorion gets tough and hard just after the fertilization or to contact with the water and provides a difficulty in injecting the DNA. Use of the following methods can solve this problem. By using the micropile, that is, which has an opening on the egg surface for sperm entry during the fertilization for inserting the injecting needle. Next, by using microsurgery for making an opening on the chorion. Then, by digesting the chorion with enzymes. Or by using 1 mm glutathione for initiating fertilization and reducing hardness of chorion. Or by direct injection to the unfertilized eggs. Another technique of gene transfer is intranuclear microinjection, which involves direct physical approach using a fine needle to deliver DNA into the cell or even nuclei. To facilitate the rate of microinjection, protoplast with partially reformed cell wall may be attached to a solid support with artificially bound substrate without damaging the cells. Solid support may be of either glass cover slips or slides. Steps for microinjection technique. First, the desired eggs and sperms are stored separately at the optimum conditions. Add water and sperms and initiate the fertilization. Then 10 minutes after the fertilization, eggs are decorionated by trypsinization. Then fertilized eggs are micro-injected with desired DNA just within a few hours of fertilization. DNA is released into the center of the germinal disc to the first cleavage in decorionated eggs. The time available for microinjection is first 25 minutes and that too between fertilization and first cleavage. After microinjection, the embryos are incubated in water until hatching takes place. Advantages of microinjection technique. Optimum quantity of DNA can be delivered per cell, increasing chances for integrative. The delivery of DNA is precise even into nuclei of target cell, again improving chances for integrative transformation. The cell structure can be injected, the small structure can be injected. It's a direct physical approach. Hence, it is a host range independent. Now, if you look into the disadvantages of microinjection technique, a single cell can be injected at a cell. Hence, it is time consuming process. It requires sophisticated instruments and specialized skills. Limited embryonic time restricts injection to more eggs and a low transformation rate. Then, if you see the picture, we can clearly understand how the micro injection procedure has been done. After that, how the fish is growing into a different size and different visibly also it looks different. This one is a transgenic and this is non-transgenic. Transgenic ones are having added colors, whereas non-transgenic, no color. Increased growth and size growth hormone gene inserted into fertilized egg first. Then transgenic salmon grows about almost 10 to 11 times faster than normal fish. Then glowfish. GM freshwater zebrafish produced by integrating a fluorescent protein gene from jellyfish into embryo fish. Next, the second method is gene transfer by electroporation. It's a simple, fast, efficient, and convenient method for transferring genes. This method involves an electric pulse to deliver DNA into cells. The cells are exposed to a short electrical shock. The cells are exposed to a short electrical shock, which make the cell membrane temporarily permeable to DNA. The desired DNA fragment is placed in direct contact of protoplast membrane, which enters into the cell upon electric shock. Hole may be created as a result and stabilized by a favorable dipole interaction with electric field. Electroporation involves a chain of electric pulses for for permeation of cell membrane, thereby allowing the entry of DNA into fertilized eggs. The rate of DNA integration in electroporated embryo is more than 25%. More than 25% is the survival rate, which is slightly higher in comparison of micro-injected ones. This is how the electroporation process will be done to extract the genes. Next is antifreeze protein gene transfer, APGT. APGT. Many teleosts inhabiting icy marine water in the polar regions produce antifreeze glycoproteins or antifreeze proteins in their sera to protect them from freezing. This protein lowers the freezing temperature of solution without alerting its melting temperature. Thermal hysteresis, the difference between the freezing and melting temperature. Despite their similar antifreeze properties, these proteins are quite different in their protein structures. There are one type of AFGP and three types of AFP. Recently, fourth type of AFP has also been reported in longhorn sculpin. The Atlantic salmon, Salmonella, lacks any of these or AFP's gene and are unavailable to survive in sub-zero seawater temperature. And inability to tolerate temperature below 0.6 degrees centigrade to 0.80 centigrade is one of the major problems of sea cage farming in northern Atlantic coast. Hugh and his co-workers developed antifreeze resistant Atlantic salmon containing an inability to tolerate temperature below minus 0.6 degrees centigrade to minus 0.80 centigrade is one of the major problems of sea cage farming in northern Atlantic coast. Hugh and his co-workers developed antifreeze resistant Atlantic salmon containing the AFP or AFGP genes using gene transfer technology. Coming to growth hormone gene transfer. Recently, scientists have developed an all fish growth hormone model. They have cloned and sequenced the grass carp and common carp, carbonic anhydrase gene, and growth hormone gene. The grass CA gene promoter has been linked to a grass carp growth hormone to form a high efficiency expression vector called PCAZ. Using the CAT gene as receptor gene, a PCA grass carp growth hormone was micro-injected into fertilized, non-activated common carp via the micropyle, generating all fish transgenic carp. The presence of transgene was detected by reverse transcriptase and northern blotting. These transgenic fish showed about 137% high growth rate of control. Now, disease resistance gene transfer. In China, scientists piloted a gene contributing resistance to the grass carp hemorrhage virus. 11 different gene fragments encoding protein was cloned and isolated from translation in vitro using GCHV genomic single gene fragments. Based on the information of capsid protein, SP6 and SP7, gene cDNA, three oligonucleotides were synthesized and fused with SV40 MT promoter and transferred into grass carp cytokine induced killer cells via a constructed expression vector and transferred with GCHV. The result indicated that the moralities were reduced by one order after challenging with the virus. Then coming to applications of transgenic fish. Environmental concerns about transgenic fish. The primary environmental concerns about releases of transgenic fish, for example, include competition with wild populations, movement of the transgene into the wild gene pool, and ecological disruptions due to changes in prey and other niche requirements in the transgenic variety versus the wild populations. Transgenic fish could threaten wild populations. West Lafayette Purdue University researchers have found that releasing a transgenic fish to the wild could damage native populations even to the points of extinction. Transgenic fish could present a significant threat to native wildlife. Transgenic fish are typically larger than the native stock, and that can confer an advantage in attracting mates. Moyer says if, as in our experiments, the genetic change also reduces the offspring, our ability to survive and transgenic. animal could bring a wild population to extinction in 40 generations. Although a Canadian research facilities elaborate precautions are being taken to prevent the release of transgenic fish into the environment. The fish are often raised in ponds covered with nets to keep birds out, enclosed by electric fences to keep muskrats, raccoons, and humans out, and the outsiders are fitted with screened drains to prevent the loss of small fishes or eggs. Next is gene flow. One of the larger environmental concerns raised by transgenic fish is the possibility that a transgenic fish species raised in open with water pens will escape and spread novel traits into the journey of breeding and wild breeding. Gene flow between the concept which factors in characteristics such as the juvenile and adult viability of a fish, the number of eggs produced by a female, and the age at which a fish reaches sexual maturity provides a usual barometer for discussing some gene flow scenarios. According to one scientific model, if a transgenic fish escapes and mates with a wildlife, gene flow could follow one of the three scenarios. One is purge scenario. When the net fitness of a transgenic fish is lower than that of its wild relatives, naturally selection will quickly purge from the wild population any novel gene introduced by the transgenic fish. In theory, evidence of the novel trait will disappear from subsequent generations. Next, next is spread scenario. When the net fitness of a transgenic fish is equal to or higher than the net fitness of a wild mate, gene flow is likely to occur and the genes of the transgenic fish will spread through the wild population. This means evidence of the transgenic genome would persist in subsequent generations. Then the third one is Trojan gene scenario. When the net fitness of a transgenic fish is altered such that the fish has enhanced mating success but reduced adult viability, that is, chance of surviving long enough to make. Then introduction of that fish into the wild population could result in a rapid decline of the wild population. Essentially, mating success would ensure the spread of the novel gene throughout the population, but the inability to survive would essentially mating success would ensure the spread of the novel gene throughout the population, but the inability to survive would reduce the population size of subsequent generations and potentially lead to extinction. Transgenic fish invasive species. Even if they do not breed with wild relatives, transgenic fishes that escape into the natural ecosystems could be an environmental nuisance by becoming an invasive species. This danger mainly arises for those transgenic fishes endowed with new genes that improve such fitness traits as breeding capabilities and the ability to withstand harsh fish or harsh conditions. The establishment of a thriving transgenic fish population in an ecosystem where it has never existed could crowd out native fish populations. Then, risk mitigation. It is important to note that developers of transgenic fish are attempting or reduce or eliminate both new genes and invasive species like risks by sterilizing transgenic fish. Sterilization is relatively easy and inexpensive, but success rates are highly variable. In addition, sterilization does not necessarily neutralize environmental risks. Academic scientists note that an escaped sterile fish might still engage in courtship and spawning behavior, disrupting breeding in wild populations. Waves of escaped sterile fish could also create ecological disruption, as each group is replaced by another equally strong group of transgenic sterile fish. Then, food safety issue. One important food safety issue involves the extent to which fish absorbs and stores environmental toxins such as mercury, high levels of which could pose a danger to humans who eat the contaminated fish. Some scientists worry that discrete biological changes introduced by the genetic engineering process might enable transgenic fish to absorb a toxin that conventional fish cannot absorb, or to better tolerate higher levels of a toxin already known to cause concerns. Some scientists have expressed concern that the genetic engineering process could increase increase the allergic potential of fish, particularly through the introduction of lower proteins that never before existed in food fish or in food chain. Biosynthetic growth hormone and growth enhancement application in biotechnology. In recent years, growth hormone, cDNA, and genomic DNA have been isolated and characterized for several species. Expression of rainbow trout or striped bass GH in E. coli cells results in production of large quantity of recombinant GH polypeptide because the GH polypeptide is highly hydrophobic and contains more cysteine residues. The newly synthesized recombinant GH, the newly synthesized recombinant GH polypeptide forms insoluble inclusion bodies in E. coli cells, rendering the hormone inactive. For activation of the hormone, a buffer containing 8 molecules urea and మళ్ళీ ఆడ then 8 urea and renaturing the polypeptide by slowly removing the urea from the protein solution is carried. In vitro application of recombinant GH to yearling rainbow trot resulted in a significant growth enhancement. In 4 weeks, weight will be doubled. In rainbow trout fry or small juveniles, immersed in GH growth promoting effect was achieved. Further application of trout recombinant to spats of juvenile oysters by dipping method increased shell height, shell weight, wet weight, and dry weight also. Let us just look at the summary of this chapter. Transgenes in many transgenic individuals are also transmitted through the germline to subsequent generations. These transgenic animals play important role in basic research as well as rapid biotechnology. A transgenic fish is one that contains genes from another species. Using the gene transfer technology, scientists now have created a genetically engineered variety of Atlantic salmon that grow to market size in about 18 months. Otherwise, the fish takes about 24 to 30 months for becoming market size fish. Development of transgenic fish has focused on a few species, including salmon, trout, carp, tilapia, and a few others. Faster growth is usually accomplished by transferring a fish growth hormone gene from one species of fish into another. The genetic material was injected into fertile carp eggs with microinjection. A single female can produce several hundreds or thousands eggs and thus provide a larger number of genetically identified eggs or identical eggs. The most commonly used methods in fish biotechnology are chromosome manipulation and hormone treatments, which can be produced triploid, tetraploid, haploid, gynecogenetic, and androgenetic fish. Now let us see the questions that are asked frequently. Model examination questions. Write about development of transgenic fish. Write in detail about gene transfer technology for the development of transgenic fish. Then write about application of transgenic fish. For short answers, write a note on transgenic fish, write a note on transgenic salmon, medaka fish or zebra fish. Then write a note on autotransgenesis, controlled culture of transgenic fish and feed, green transfer of micro injection, the gene transfer of micro injection. Write a note on steps of micro injection technique. Write a note on gene transfer by electroporation, growth hormone gene transfer. Write a note on disease resistance gene transfer. Then write a note on gene flow. Then finally, write a note on biosynthetic growth hormone and growth enhancement. So with this, we have come to the end of the chapter. Hope you all have understood. Thank you.